Hello everyone, this is the next video lecture on sexual reproduction in flowering plants. And in the last video lecture we studied about double fertilization and along with that artificial hybridization. In artificial hybridization, emasculation and bagging definitions are very important. Please remember, emasculation, the chance of asking question on emasculation is very high. So please remember the definition of emasculation. It is nothing but the removal of the anther from the flower. Okay, that is emasculation. And after that, double fertilization. Double fertilization is also very important and very simple concept. And they will ask the question, explain the double fertilization. What is double fertilization and how the double fertilization occurs? Everything we studied in the last class. Okay, here what is the process? When the pollen grain or fall on the stigma, the generative cell start germinating, and then finally it sends the pollen tube towards the microfilament, and in the microfilament synaptic cells are there. Those are going to be fused with that, and those are going to be synaptic cells are there. They will guide the gamete for the fertilization, and one of the gamete is going to be fused with egg cell, another one is going to be fused with polar nuclei. Okay, this is the double fertilization. And moving to the next concept, that is post fertilization. Okay, those are all that what we studied. Double fertilization is fertilization events. Okay, and before that, the formation of the gametes, it means the formation of the pollen grain, formation of the ovule, everything. Those are all the uh, pre-fertilization events. Okay, those are all the pre-fertilization events. But after that, the fusion of the gametes or syngamy is the fertilization event. And after the fertilization, what will happen? That is post-fertilization events. Series of events we can see in case of post-fertilization. Total 4 are going to be happen here. One is development of endosperm. Second one, development of the embryo. Third one, maturation of ovals into seeds. Next, ovaries into fruits. Okay, first what will happen? There is a formation of endosperm. Next to that, there is a development of the embryo. Next to that, there is a ovules are going to be formed into seed, and then ovary is going to be to form a fruit. Okay, this is the thing. Please remember, in the last year they asked the question, name the part of the plant which is going to be developed into fruit. The answer is ovary. Ovary is going to be developed into fruit, and ovule is going to be developed into seed, and fertilized zygote is going to be developed into embryo, and triple fusion, that is polar nuclei, those are going to be developed into endosperm. Okay, these are all going to be happen in post fertilization events, and we'll study one by one. Okay, listen, next to this one here you can see the endosperm. First one is the development or the formation of the endosperm. Why the first endosperm is going to be formed? Okay, during the process or during the maturation and development, antipodal cells are providing the nourishment to the embryo sac, complete embryo sac. And after fertilization, what will happen? Embryos in the embryo sac, antipodal cells and synergid cells are going to be degenerate, or they are going to be disappear. So when the fusion occurs, there is a formation of the zygote. So the zygote need energy or ATP. Zygote need energy or ATP, and that energy is mainly provided by the endosperm. Endosperm will give the nourishment to the developing embryo. Endosperm will give the nourishment to the developing zygote or developing embryo. So for that purpose, first there is a formation of the endosperm, not the embryo. Okay, first there is a development of the endosperm, and this endosperm will give the nourishment to the developing embryo. Okay, this is the concept. And if you observe the endosperm, Okay, it is nothing but the polar nuclei. They are going to be, sorry, polar nuclei, they are going to be formed here. These two polar nuclei are there, and these two polar nuclei along with one egg cell, there is a fusion, and this is fusion is triploid. Okay, this fusion is triploid, 3n. Okay, but the zygote is 2n. Okay, already two cells are there. Along with these two cells, there is another male gamete is going to be fused, so it becomes 3n, that is triploid. But the zygote, one cell, there is a fusion of another male gamete, it becomes diploid. Two cells are going to be formed. Okay, that we'll study next later. And here, if you observe the endosperm, okay, here the given uh, endosperm gives the nourishment or nutrition to the developing embryo. These endosperm cells mainly contain nutritions. Okay, they are nutritious in nature. It depends on different species. Okay, either they are mainly contain starch, they mainly contain oil, they mainly contain protein. Okay, uh, different nutritions they have. And if you take the example for protein, we can take maize. Okay, endosperm richly contains protein. And if you take the rice, rich amount of carbohydrate. Okay, like that there is a different nutritions are present in case of endosperm cells. And here during the development of embryo, there is a nutrition. Okay, there is a requirement of the nutrition for the developing embryo, and that is provided by the endosperm. And here, if you observe the endosperm, there is a two types of endosperm we can see here. Okay, in the beginning, what it is? It is PEN, that is polar endosperm nucleus. Okay, polar endosperm nucleus it is. And this polar endosperm nucleus is undergoing division. It undergoing lot of divisions, repeated divisions. Based on that, we are going to be divide here the endosperm into two types. One is prenuclear endosperm, another one is cellular endosperm. Please remember, two endosperm types of endosperms are there. One is prenuclear, second one that is cellular endosperm. Prenuclear endosperm and cellular endosperm, two types are there. Let us see what is prenuclear endosperm. What is meant by prenuclear endosperm, that I will tell you in this slide. This is a structure. You can see the embryo sac. That I will come later. And here I will show the prenuclear, first one, that is prenuclear endosperm. What happens in prenuclear endosperm? Listen, this is the one polar nuclei. Okay, primes are not polar. This is the primary endosperm nucleus. Primary endosperm nucleus. In pre-endosperm nucleus, what happens? There is a division of the nucleus, not the cell. Please remember, there is a division of the nucleus. For example, this is the primary endosperm nucleus. What will happen? In this, the same thing here. This nucleus, whatever the nucleus present in the middle, that is going to be divided. That is going to be divided. The nucleus, those are present here, they are free. They are free, they don't have any cell, they don't have mainly covered, they are not covered by the cell wall. If there is a covering of the cell wall, then it is, we consider it as a cell, but there is no covering of the cell wall. The nucleus are free here. Such type of endosperm we call it as a pre-nuclear endosperm. Okay, pre-nuclear endosperm, we are going to call it as a pre-nuclear endosperm. And this we can see in case of coconut we can see here. Okay, in coconut we can see the endosperm, this prenuclear endosperm. The middle water is present in the coconut. Okay, in the middle we can find the water. That water is nothing but prenuclear endosperm. Please remember that. pre-nuclear endosperm it is. Okay, in the middle coconut, in the middle of the coconut we'll find the water, and that water we call it as a pre-nuclear endosperm. Please remember, okay, for your competitive exam also it is very important. That is, in competitive exam also it is very important, please remember. Okay, this is about the pre-nuclear endosperm. I think you understood about this. What will happen here? The middle nucleus, the single nucleus is going to be divided multiple times. Okay, this triploid nucleus is going to be divided multiple times, and there is a formation of the pre-endosperm nucleus. There is no development of the cell wall, only there is a division, only there is a karyokinesis, not cytokinesis. Only karyokinesis occurs here. Okay, that is karyokinesis, not cytokinesis. Okay, this is the pre-nuclear endosperm. And let us see in the next one, that is cellular endosperm. Sorry, next to that here you can see cellular endosperm. In cellular endosperm what will happen? Say for example, this is the primary endosperm nucleus, primary endosperm nucleus, and here primary and endosperm nucleus will undergo division. One cell become two, and 2 it becomes 4, 2 it becomes 4. Okay, only the nucleus is not going to be divided here, complete cell is going to be divided here, complete cell is going to be divided here. There is a development of the cell wall we can see in case of cellular endosperm. Development of cell we can see here, cell wall we can see here. Okay, like by doing like this there is a formation of lot of cells, there is a formation of lot of cells like this. You can see a number of cells are going to be formed here. Okay, a number of cells are going to be formed here. This is polar endosperm tissue, polar endosperm tissue, because cells united to form a tissue, so it is polar endosperm tissue. Okay, please remember students, there is the presence of two types of endosperm. One is pre-nuclear endosperm, another one is cellular endosperm. In pre-nuclear endosperm only the nucleus is going to be divided. Okay, karyokinesis occurs. Okay, it occurs in case of coconut. And coming to the cellular endosperm nucleus, here the complete cell is going to be divided. 1 become 2, 2 become 4, 4 become 8, like that. There is a development, and there is a formation of polar primary endosperm tissue. Okay, this is also we can find in case of coconut. In coconut we can take the example, both free nuclear endosperm is also present here, cellular endosperm also present here. That white portion we can see in the coconut. Okay, if you think the white structure, okay, if you observe the white structure in the coconut, okay, this is the outermost structure of the coconut, and in next to that we can find the white portion. Okay, you can find the white portion. That white portion is a cellular endosperm, that white portion is cellular endosperm, and in the middle we can find the water. This water is pre-endosperm nucleus. This water is free endosperm nucleus, not cellular. Okay, it is a free endosperm nucleus, and that white part we call it as a free cellular, free endosperm nucleus. Okay, free nuclear endosperm we call it as. Okay, this watery part, and white one we call it as a cellular endosperm. Okay, I think you understood about these two concepts. One is free cellular endosperm, another one is cellular endosperm. Okay, next to this, in some seeds what happens, the endosperm is going to be consumed completely. Okay, in some cases the endosperm is going to be consumed completely, or in some cases what happens, it retained, or it sometimes it is not completely consumed. Okay, so a small portion of endosperm is going to be remain over there. Okay, some small portion of endosperm is going to be remain over there.
Okay, based on that we are going to be call if the endosperm is completely consumed. Okay, so when the seed is formed completely, at that time there is no endosperm. Such seeds we call it as a non-endospermic seeds. Non-endospermic seeds. If the endosperm persist, if the endosperm persist, such we call it as endospermic seeds. Such we call it as endospermic seeds. Such seeds we call it as a sorry such seeds we call it as a endospermic seeds. Okay, it do not contain the endosperm, those are non-endospermic. If they contain the endosperm, those are endospermic seeds. Okay, for non-endospermic seeds we can take it as example pea and then beans. Groundnut also comes. Okay, pea, groundnut and beans, they all comes under the non endospermic seeds because they do not have any endosperm. The endosperm is completely consumed by the embryo, developing embryo. Okay, and during the germination there is no endosperm, and it is some cases, in some cases it is persist or it is retained. Okay, when it is retained during the mature seed. Okay, in the mature seed it is going to be retained. And here you can take the example coconut and castor. Coconut and castor are the example for endospermic seeds because they contain endosperm. They contain endosperm in that. For example, you can take coconut. Okay, there is a presence of cellular endosperm and then there is a presence of prenuclear endosperm. Both are going to be persist for a longer time. Okay, when this endosperm is going to be utilized during the germination of the seed. During the germination of the seed, that persistent endosperm is going to be consumed. Okay, that persistent endosperm is going to be consumed. Okay, that is going to be utilized by the seeds for the further germination and everything. Okay, like this there is the endosperm. Okay, this is about the endosperm. Please remember the prenuclear endosperm and then cellular endosperm, types of the endosperm. The main function of the endosperm, it mainly nourish the developing embryo. That is very important. It will nourish the developing embryo. Please remember that. Okay, it is very important. Please remember. And this is about the endosperm. Okay, this is about the endosperm. And in the next video lecture I will begin with uh, embryo. Okay, development of the embryo. Okay, how the embryo is going to be developed. Okay students, what we studied today, we studied about the endosperm. Okay, in the next video lecture we'll move to the next concept, that is embryo, development of the embryo. Okay, thank you.